Hi friends, now we are discussing about UV visible spectrophotometer. First, we discuss about what is spectroscopy. Spectroscopy is a branch of science that deals with the study of interaction of matter with light based on the principle Beer-Lambert's law. Okay, now we are discussing about UV visible spectrophotometer. In UV visible spectroscopy, we use UV light and visible light. Then, UV visible spectroscopy is also called as absorption spectroscopy or electronic spectroscopy. And there is a common solvent used to preparing sample is either ethyl alcohol or hexane. Now we are going to instrumentation of UV visible spectrophotometer. In first, we discuss about what are components used in UV visible spectrophotometer and what are their function. First one is source, and second one is monochromator. Third one beams filter. Fourth one pair of identical cells. Fifth one detector. Sixth one recorder. Then what are their functions? First, source. Source consists two components, hydrogen lamp and tungsten film lamp. Hydrogen lamp to produce ultraviolet rays and a tungsten film lamp to produce visible rays. Second one, monochromator. Monochromator allows the chosen wavelength and absorbs all wavelengths. Example, prism. Third one, beams filter, which splits the beam of light of each wavelength into two equal halves. One goes to the test sample and other to the blank or reference sample. Fourth one, pair of identical cells. One cell is filled with the test solution and another with solvent. Example, quartz. Fifth one, detector. Detector detects the intensities of the transmitted light coming out of the cells and compares each wavelength and generates a current proportional to the difference in the intensities of the sample and blank. Example, photocell and photodiode. And last one, sixth one, recorder. It records UV visible spectrograph between wavelength and observance. Now we go to the procedure. First, source releases ultraviolet rays from hydrogen lamp and visible light from tungsten film lamp. And both lights are with different wavelengths. Now monochromator. Monochromator is used to absorb all other wavelengths and allow only the chosen wavelength. And then splitter. Splitter it splits the light of each wavelength into two equal halves. One goes to the test sample and other to the reference sample. And there are a pair of identical cells. In those cells, one cell is filled with test solution and another with solvent. And then detector. Detector it detects the intensities of the transmitted light coming out of the cells and compares each wavelength and generates current proportional to the difference in the intensities of the sample and reference cell. And finally, recorder. Recorder it records the UV visible spectrograph between two components, wavelength and absorbance. And finally, we discuss about what is the use of this experiment. In this experiment is used to identify the chemical composition of matter and to determine the physical structure.